Why are you pushing our partners?
So we are going straight to the lab. We do the functional opening. Yeah. Yes.
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 My team, chairman, CEO.
Okay.
Yeah. The microphone, you can read.
You can read it? Okay. Thank you. Yeah.
I'll ask our lab manager to see how the workflow goes.
This again strengthens Kenya's leadership not only in the Africa region because it's a major reference lab. You will see when we start talking, actually there are a number of countries that are already using this facility that rely on Kenya. And most importantly, now you no longer need to send it to CDC Atlanta. The turnaround time was about 35 days. Now with this, it's going to at least half. So the investment is roughly about $500,000. So that's okay.
Thank you. John. John, he has got to pay your bill.
For confirmation of polio. But now everything is going to come here. This is a special lab. We support both the Afro and the EMRI region. So we are working for 6 countries. And we have a capacity of 21 people working in the polio workflow. And they do different activities. So this particular facility that has been brought in is not only reducing the turnaround time, it's going to tell us whether we're having a wild polio virus, vaccine derived, whether it's having a mutation, so that the Ministry of Health can have time to have a response before we go out of hand when it comes to polio diagnosis. So when you look at
you can enter this particular lab
yes,
only?
yes. When you look at this lab, we have state-of-art machineries. Please come in.
Allow her.
Thank you. But we are having others that are doing targeted what goes on here.
Welcome our visitors. This is the polio genomic sequence.
The people of the cameras take photos and then go out. You want to listen to what we are being told. Yeah. We have too many cameras and the staff.
For sequencing, we have the Sanga sequencer. This is the SEP studio for fragment analysis. We have the Oxford Nanopore for fragment analysis. However, we have the Illumina ISEC for whole genome, and we have the other Illumina for also whole genome sequencing. So basically what we do here, we sequence pathogens. This one is used for polio and measles sequencing platform, and also the Nanopore is for polio and measles, while the MySec is used for multi-pathogen detection. So maybe it's one kind.
Yes, so we are not only doing polio, we are also having a polio legacy where we are testing more than just polio because we work on environmental samples. And for these samples, they have a lot that's coming in. You know, most of the times you don't have diagnosis in the community because a lot of people go to the chemist. But when you check in the environmental samples, you can know what is ailing the community. You can actually know issues of antibiotic resistance. So that is what we check besides polio and measles.
The idea is
We have sewer. We do sewer. Yes, we do sewer. Yes.
So the idea is when you set up the infrastructure and the equipment, so as you said, you don't focus only on one disease. You set the capacity to process also all pathogens of epidemic and pandemic potential. So beyond the polio, and that's the whole idea.
Absolutely.
So is this where we did the mpox sequencing or?
Yes.
Yeah.
Yes.
And was really we did the vaccination. We utilized all the vaccines for mpox.
Yeah. Even the last response we had for measles, the results came from here. Yes.
We're going to tell the Waziri the countries that are supported from here,
Yes.
so he hears them.
So we are supporting Kenya, being a Kenyan lab. Then we are supporting Djibouti. We are supporting Yemen, Comoros. We are supporting Eritrea. We are supporting also Somalia,
Ethiopia.
Yes. So we support 6, 7. Yes. Yes.
You cover the whole of East and
7
Yes. All the other countries in Afro just support Afro, but we support Afro and EMRO. Yes.
EMRO is countries in our Eastern Mediterranean region in addition to Africa.
Yes.
Good. Thank you, John.
And we also have measles and rubella database. So where we project here and do QC and interpret to the ministry to write a small narrative of what the virological outbreak actually means to the ministries. And then our honorable Dr. Ahmed will now mount a response depending on what we have seen here. So why we have such a platform is to do QC. Because one eye, quality control, so that we can have the same interpretation of the outputs of the virological analysis.
So if I want to summarize this, I would say 3 things. From lab to data to policy.
To policy, yes.
absolutely those three. So the data must be right.
Yes.
So the policy to be solid.
Yes.
And the team must be like not less than 5 persons to say, to write a small one. In concurrence with reference notations of Afro and NICT and
So for this particular area, this floor, we call it the wet labs. Because this is where all the reagent and the samples are handled. The workflow begins from there, coming this way. This way you visualize. What have you brought out from your experiment? You'll see from here through gel electrophoresis. We have a state-of-art imager that shows you the output of your work. Yes. We have a shadow from my side.
You're the bonus one.
No, you are safe. That's where we safe.
Yes,
This is where you get your DNA magnified to many copies, the ones that you need. And from here, you get to pick out your DNA of interest. This is a target for polio. You pick it out, amplify it, make it big. Then you go downstairs to analyze it further to know exactly. Because at the end of the day, the type is very important. For the program, it's not just all polios that give us panic. Type 2, when it's mutated, we all panic. Wild type, we all panic. Hapa ndio msema kweli. We'll tell you whether you have it or not before you visualize it. Yes. This is why we prepare the sample before it goes now into the analysis for PCR. Okay, and sequencing. So this preparation, and this is the clean room. This place does not have any virus. It's clean reagents that is used downstream. So if you do a mess here and contaminate the work, everything in the workflow is completely out. Yes. And that would be the end of the tour of this. We just want to connect you.
Well, so sir, this is basically the beginning of the workflow. Yes. So we use cultures to grow viruses. So viruses do not grow on any other medium apart from cultures. So you can see all these flasks. We inoculate these flasks because they contain cells, and we grow cells here. Also, a lot of the support equipment, including having our own water, distilling the water and so on, requirements for this particular place. So the work starts here, and we go. One of the only three in the country. Yes. In very, very simple language, it basically means you can handle high consequence pathogens. Yeah, you can handle Ebola, you can handle Marburg in this particular facility.
So in another word, yes, labs are put at different levels based on the severity, the seriousness of the type of germs you're dealing with. The P3 lab is, let me just say, it's a senior lab. It's a very high security
flow. From here, basically, this is general virology, going in this direction. A lot of molecular procedures happen in this, like gene extraction, post-PCR and PCR activities on this particular area, viral hemorrhagic fevers. So they help the country in terms of ensuring that EBDs and other Ebola virus disease, Marburg and so on, and yellow fever, and they are very active. So Dr. Gonongo,
those are called the hemorrhagic fevers.
Hemorrhagic fevers. So they handle them here. So we'll not be able to go here because it's highly dangerous. And then this other section now is for human samples, so polio viruses. So this is AFP, you can see acute flaccid paralysis. So here we actually treat samples, and the next one we amplify the samples by growing them. So basically here on this particular space,
this is happening in children.
yes, acute flaccid paralysis. Because when you are doing polio surveillance, you don't look for polio, you look at a syndrome, right? So, and you can see that the colleagues who are working in the safety cabinets, these are very protected cabinets that really help them secure the individuals. Right? And for this section, so this is human. This particular section from here to the next laboratory, we have environment. So we process basically sewage, we process wastewater, that, and we analyze for polioviruses.
So it's very simple. Sample is taken from sewage water, from the environment. You take it there, you bring it here to analyze. So basically, it's just from the experiments.
Yes.
So you take it, you come, you analyze it. That's what the environment has.
And historically, so when we started this laboratory, the first sample we took, like WR says, the first sample we took, we got a wild poliovirus. Imagine. And now this is the big story, the last one basically. The big story basically about the connectivity of the workflow. So this is the space where we used to do all this molecular work that we are now transferring on this other side. So this space was very, very restricted in terms of really trying to work and look at all the procedures that we are transferring to the other side. So the capacity in terms of space, in terms of configurations and so on, could not enable us to do up to sequencing level. So it then limited us to actually sending samples out of the country, this particular facility, this one. But now that we have the bigger facility, yes, we now support it. And then we'll disinfect it. You get your hand wash. I mean, there is some sanitizer here. We wish really to thank WR and the high levels of really mobilizing, because without that kind of mobilization, we can no good.
Thank you. Yes. Do you want to thank you? Need you to be able to.
Oh yes, part of the 15 billion.
Adirwa, please take over.
Okay.
All right. So Adirwa is our director for Division of Medicine. And yes. So explain the significance of this.
Good morning, ladies and gentlemen. My name is Aden Duale. at scientific publication of and what we are requesting you to do today is to help us plant some of the plants that have great significance medicinally. One of them being this one, which is called Prunus africana. That is the scientific name, but the local name is called Oil. Yeah, and it is used for prostate. It's very good for prostate. We have done scientific validation for the same, and we have seen very encouraging results. It's also good for allergies, and so today, we kindly request you to help us plant trees.
Part of the 15 billion. This or this?
Either.
And we have found you have very good antimicrobial activity, so it can be used for as a microbial for treatment of microbe infections and fungal infections and such. So we'll welcome representatives from the W Group to plan for us.
To go. Take from here? Dr. Ahmad, you see I know how to do this, right? Eh? I don't handle only cows, eh? I don't do only cows, eh?
Yes. It's that one. This is called Orbicula eucadensis. In Peru, it's called surrogate. Yes, it's an antimicrobial as well. Yes, and it's also good for toothaches. Yes.
For toothache, eh?
Yes.
Oh, because it's
welcome.
okay.
So I will have
I have a few fish spots.
Oh, the other side. Okay.
Livestock keeper.
You know trees. Yeah, no, Dragon was talking about cows and now we're telling the fish. They don't know how to build the house.
Sorry, this is a tree that everybody knows. It's called Azariata indica ama mwarubaini. You all know mwarubaini.
This is a famous.
And it is acclaimed to treat many diseases. We have seen that it is very good for. Well, it treats malaria. Yeah, it also treats infections and also antimicrobial.
Yeah, if you do the skin.
Yeah, for fever as well.
Those things is okay for that.
This is the executive school. Where we are now going. You have to not only have. Yeah, those are significant. work and we are doing scholarship. Different at the top. Different at the top. So this is so today we just confine ourselves to the function of the time. Okay. So we could take you. Okay. Yeah.
This pen has no ink. This pen has no ink. Where's my pen?
It's too much movement.